Hi, this is Marjan, and I'm going to talk about chromatin state annotation of regulatory elements in human cell types using Segway. Sequencing-based genomic assays can measure DNA accessibility and histone modifications. For example, throughout DNA-seq and free-seq regions sensitive to cleavage by DNase I enzymes are identified which show DNA accessibility and have been associated with various regulatory regions such as promoters, enhancers, etc. Through ChIP-seq, we can measure various histone modifications. For example, in this figure, we are looking at these measurements in a specific DNA region for a biological sample. Each row demonstrates the signal for a specific histone modification and is a result of a sequencing assay. We call each row a track here. For a single biological sample, we can do multiple sequencing experiments to get these signals. Various combinations of these signals can highlight different regulatory mechanisms. For example, a specific combination of signal from these three tracks modifications can signify a promoter and active enhancer regions in the genome. Essentially, by analyzing these signals from different tracks for a cell type or a tissue, we can obtain a clear picture of its genomic functional elements such as active enhancers and promoters. Multiple epigenomics data sets on various cell types and tissues have been collected by ENCODE. Our goal here is to obtain chromatin state annotations resulted from all available data from ENCODE. That will result in chromatin state annotations for hundreds of cell types and tissues. The complete data set of all the available cell types and their epigenomic assays could be demonstrated in a matrix like this. For an example, we can consider the process of chromatin state annotation for one cell type. Segway performs the task of segmentation and genome annotation in two steps. In the first step, Segway performs unsupervised high-resolution partitioning of the genome into several chromatin states based on the input epigenomics tracks. It assigns each segment to a state label corresponding to a type of genomic activity. In the second step, an automated process assigns human understandable interpretation terms to each state label, such as quiescent, enhancer, and promoter. This is done based on the features of the state label, such as average track values throughout the genome and their placement around the genes. As I mentioned, we use label features to determine their interpretation terms. We first do it manually for a handful of samples, which are used as training examples for a classifier. The classifier is then used to determine the interpretation terms for the rest of the samples. This allows to perform the interpretation process efficiently for a large set of samples that we have in this project. The main product of this project is a set of high-quality chromatin states annotation for all the available human epigenomes from the ENCODE. A direct application of such annotations is in understanding the regulatory role of genomic variations such as GWAS variants. Please come and see the poster for discussions on the current and future work in this project.